Welcome back to the channel. Today we'll be talking about COVID-19. Most viral vaccinations consist of injections of either dead or attenuated viruses. Dead viruses are the best for vaccination as far as establishing a broad immune response. The drawback is the limited amount of dead viruses that you can inject, hence a limited immune response. Another type of vaccine is what's called a live or attenuated vaccine, which contains a version of a live virus that has been weakened so that it does not cause serious disease in people with healthy immune system. It can cause disease though, just not serious, hopefully. Attenuation and weakened are very deceptive words in this context. In this case, they mean that through successive generations of cell lines in foreign hosts, the wild virus has mutated and is now less virulent. Some of these cell lines started in 1959. This mutated virus is not identical to the wild virus you are attempting to induce an immunity to, but close enough to be effective. But these attenuated viruses are still replicative. They will enter cells and start reproducing, but the virulence has been attenuated. The viral load is small, and the body normally can stay well ahead of the infection. Every cell a virus enters will ultimately be killed by the virus. Let that sink in. However, a live attenuated vaccine may still cause severe or fatal infections as a result of uncontrolled replication of the live attenuated vaccine virus. However, this normally only occurs in persons with a weakened immune system, but not always. Dead virus vaccines do not have this problem. With live attenuated vaccines, we get an amplification effect. We can inject a few viruses. They replicate and produce a lot more viruses, which gives the immune system a bigger target to respond to. A virus cannot enter a cell except through a receptor. Once a virus enters a cell through a receptor, it hijacks the nucleus and tricks the cell into making copies of itself. The cell will continue to make copies as long as it can. These copies remain in the cell. Eventually, the cell dies from this process. A process of macrophagy occurs where the cell is broken down. When it is, these manufactured viruses are released into the lymph and reinfected other cells, and the process repeats until the body mounts a sufficient immunological response to stop it. The same process is true for the attenuated virus. You again can see the amplification factor in this process. One attenuated virus enters a cell and hundreds or thousands are manufactured. This results in a stronger immunization response. The incubation period is a time from when the cell is first infected and when it dies and disperses the new viruses at such a level that symptoms are detectable. This will occur after many reproductive cycles. The downside to attenuated viruses is that they are replicative, which means you can lose control of the process. To solve this problem, someone decided to use mRNA to produce proteins close enough to those on the target wild virus to elicit a satisfactory immune response. mRNA is not capable of replicating itself, which is an improvement. However, this immunity is very narrow and not as robust as one developed with the wild virus, i.e., natural immunity. Viruses mutate constantly and rapidly, so it is highly likely the wild virus will mutate so as to make the mRNA immunity worthless. With COVID-19, a mRNA was designed to grow a spike protein that closely, but not exactly, matches a spike protein on the COVID-19 virus. This creates a very narrow immune response. If the wild virus mutates its spike protein, then the vaccine is worthless. mRNA cannot exist in the lymph, so it must be delivered in a nanolipid, which hides it from the immune system. This nanolipid is so small that it does not need a cell receptor to gain entry into the cell, like viruses need. It can just pass right through the cell wall when it comes in contact with a cell. It is just like a Cracker Jack box. It has a surprise inside. When the mRNA reaches the cell nucleus, the cell relentlessly produces spike proteins. There is no stop order. Just like being infected with a virus, the cell keeps making spike proteins until it dies from the uncontrolled manufacture of the spike proteins. In essence, an mRNA is a non-replicating virus, but much smaller. Like a virus, the mRNA will eventually kill every cell it infects. You have probably heard that mRNA causes a cell to express the spike protein. Sounds completely harmless. It's a lie. It kills the cell. COVID-19 can only infect cells with ACE2 receptors. COVID nanolipid encased mRNA can easily enter, read in fact, any cell it comes in contact with. Okay, let's look at the medical malpractice in the current program. The vaccine label clearly states that the vaccine serum is to be injected IM, or intramuscular. This is to prevent the serum from being transported throughout the body. The immune response will be localized in a large muscle that can well tolerate the immune response with minimal reactions and discomfort. However, most countries no longer require that practitioners aspirate needles before injection. However, the label requires IM. The only way you can comply with the label is to aspirate the needle. Aspiration prevents the serum from being injected directly into the circulatory system. So if you are not aspirating, then you're not in compliance with the label. That's medical malpractice. The effects of injecting the serum directly into the circulatory system range from nothing to rapid death. Just review the VAERS system to see how many millions of serious adverse effects have occurred. More people have been injured by the vaccine than were injured by COVID-19. Remember, the nanolipid can easily gain entry into any cell it comes in contact with, and the bloodstream reaches every part of the body. So if the serum is injected into a blood vessel, which can occur if you do not aspirate the needle, then any organ in the body can be readily infected. Hence all the myocarditis and nerve damage and other problems being reported in bears. These nanolipid encased mRNA are dangerous no matter how they are administered. Even when they are administered IM, they are so small that they make it into the bloodstream. This fact has been publicly recognized by the pharmaceuticals, even though they said it couldn't happen. All of what has been covered was and is well known to the pharmaceuticals and medical community. The medical community has completely failed society. The whole result is bizarre. There is no hope for those harmed by these mRNA therapies. The long-term effect on society is unknown, but the jump in all-cause mortality since introduction of the vaccine should not be ignored and needs to be explained by the medical community. And the pharmaceuticals have legal immunity for their actions. Enjoy your cheese while you walk through the maze. Let's talk about some conspiracies. It is clear from the DNA cleavage sites that COVID-19 was a product of gain-of-function research. This virus does not exist in nature. It was man-made. This type of research is specifically banned by congressional law, yet labs in Wuhan, Ukraine, and other unknown sites are conducting such research under indirect U.S. oversight and funding. It is called plausible deniability. Due to negligence at the Wuhan lab, this virus escaped into the Chinese population. No one knew what the effect this virus would have on humans, so the authorities went full Monty in their response. Lockdowns, face masks, quarantines were instituted. The R0 transmissibility rate on this virus is very high, and none of these efforts was effective at stopping the spread of the virus. An amazing amount of money was thrown at developing a vaccine. Within 1 year, an mRNA was developed, or so it would appear. In all probability, the mRNA was developed along with the gain of function work. After all, if you're going to weaponize a virus, you want a vaccine ready to go to protect your people. It appears that is what may have happened. As of today, the virus has mutated to the point that the mRNA-produced spike protein no longer offers any immunization. The virus is mutating faster than a new mRNA can be created. The pandemic is over. Natural immunity is far superior to immunity gained from the spike proteins. Statistically, the virus is very benign, yet the lies continue. Thanks for watching.